As usual, I forgot to watch my time on the last one, and I was just saying that I keep forgetting to tell you, and that in telophase, the nuclear envelope comes back, and so that's all I was saying. So we'll move on. Um, Now, all of these phases, prophase, metaphase, anaphase, telophase, and then cytokinesis, is regulated by controls because with regard to if in fact DNA is not replicated properly, we know that that's the definition of a mutation. Mutation then can lead to problems. It can lead to good things, but it can lead to problems too. But one of the ways that they understood these cycles was in various different experiments. So each cell in your body, for the most part, they're on different cycles within themselves. Your skin cells replicate constantly. Your nerve cells don't replicate at all, or so slowly it seems like not at all. And then in the middle you have your liver cells that don't tend to replicate unless they need to, unless there's a need for it. And so the thing with regard to regulation, this happens on a molecular level. It's actually a particular compound or compounds that are found because of research in the cytoplasm. And one of the reasons why all of this is being researched is because if we can understand normal cell cycles, then we can have a greater understanding of cancer cells. And so they've learned that actually cancer cells seem to escape all of the controls that we've learned about. This particular experiment that's being shown on this PowerPoint, and also comes from your textbook, is they took cells that they knew were in a specific part of the whole cycle. So the S cycle, remember that's when the DNA is synthesized, put into a in close connection to a cell that they knew was in the G1 cycle. The G1 cycle cell starts replicating its own DNA. So the idea is that, and then also all the way through the whole thing, you put a cell that's in the M cycle, so the mitotic cycle, the G1 cycle then goes into the mitotic cycle. The idea is that the control chemicals must be found in the cytoplasm. That's what the way that these two cells could actually communicate with each other to shift what is going on in each individual cycles. And then also understanding that the chemicals that are in the cytoplasm are in there in certain percentages in those cycles. So it never happened that a G1 cell caused an S cell to decide it didn't want to go into the S, it didn't be in the S cycle anymore. Plus also some of these cycles are all or nothing. So something can be in G1 and then go into G0, meaning it's not going to replicate. But once that replication starts, it's going to it's more or less going to continue. So that's why the G1 then becomes S1. The G1 cycle goes into S, and the G1 cycle goes into M, as opposed to the other direction. In terms of the hypothesis of the experiment, different phases were fused to form a single cell with two nuclei, and then both of the nuclei go into the same phase. The control, and this is the clock that we started this whole lecture with, is a cycle. And there is definitely checkpoints and distinct control points of this cycle. It's like a clock. There are external controls and internal controls, and specific checkpoints throughout the whole system. Some of them more is known about than others, but you have a checkpoint at G1, and that actually is the checkpoint that allows the cell to actually go through replication. As I said, there is also this idea that G1 actually goes to G0, that if that checkpoint isn't passed, that cell doesn't replicate, and some of your cells operate that way. And then you also have several checkpoints in mitosis. Those checkpoints, and it makes sense that in mitosis, if all of the chromosomes aren't properly duplicated, that would be a problem. That would be a reason why this particular cell would want to stop its mitosis. If all the cells are not attached to spindle fibers in that metaphase, that would be a time to stop mitosis. And then in anaphase, if you don't have the proper complement of chromosomes going to each of the poles, so that you have those 23 pairs that are moving, you're making sure the correct 23 individual chromosomes are moving to poles, that you stop the mitotic cycle, and basically stop the cycle and cause that cell to die. So the specific checkpoints are for stop and go signals that are received throughout this whole cycle. G1 is basically, by some standards, the most important. If a cell receives the go-ahead to replicate at the G1, it will complete the synthesis G2 and mitotic phases. If it doesn't receive that go-ahead, that's when, as I said before, the cell switches into a non-dividing state called the G0 state. So if there's a go-ahead, this cell is going to replicate. If there's no go-ahead, it's not going to replicate. And then there is understanding with regard to regulatory proteins, not all of them, but one of the regulatory proteins are cyclins and cyclin-dependent kinase. So you have a complex there, and the cyclin-dependent kinase are abbreviated as CDKSs. That activity, their concentration fluctuates during the cycle. It's controlled by the amount of cyclin, and so their concentration may vary and definitely varies. And then you also have this maturation promoting factor that triggers the cell's passage from the G2 checkpoint into mitotic phase. So let's look at this image and see what it's telling us. What you have is you have cyclin begins through the G2 phase. So begins through the G2 phase, actually, and late in the S phase, and then accelerates or becomes more concentrated between a G2 phase. It's protected from breaking down during that phase and accumulates. Then what happens? It combines with the it combines with the growth factor, the growth maturation promoting factor, and that then allows the cell to pass the G2 checkpoint into mitosis, and then the whole complex promotes mitosis by this process of phosphorylation. And then after mitosis is underway, during anaphase, the cyclin starts degrading and terminating the mitotic phase as the cell enters G1. So you have this one particular protein that's understood that depending on its peaks and valleys, and I think you have a nice graph in your book with regard to the peaks and valleys of the cyclin, how when it is peaking, then you have the mitotic phase. When it's dropping down into the valley, then you have your G1, S, and G2 phases happening. The G2, I guess, as the peak is building up again. And then internal signals, so those would be checkpoints, external checkpoints, I guess you might say. For example, if a kinetochore is not attached to a spindle, so a kinetochore was not able to capture, as I said, one of those chromosomes in anaphase, this would be an internal signal that basically that mitotic division should stop. And then also some external signals are released that allow cells to stimulate and divide. So there are some growth factors that dictate when cells grow and divide. Examples of a growth factor, this is another experiment that's in your book, is you have fibroblasts, which are the responsible for growth of in the human body cell, and when they are given this growth factor, this platelet-derived growth factor, then they go into cell division. If they are not given this platelet-derived growth factor, and this is in a laboratory environment, they don't go into division. So there are other outside chemical forces that regulate when a cell will and won't divide. So the external controls can be something that is in the environment outside of the cell, but you also have internal controls with regard to when a cell does decide to divide, and making sure that that division is normal. Other I guess processes that regulate how much division we have. You have this density dependent inhibition that that cells will divide until their space is filled. And so if in fact they start to crowd, they stop dividing. And then anchorage dependent factors, where if a cell can't adhere to something, then it won't divide. But as soon as it can adhere to something, it will divide. With regard to cancer, cancer cells don't seem to adhere to either of these or many of these factors that we talked about, but definitely the anchor dependent and the density dependent inhibition. They don't respond to control mechanisms. They don't seem to need any growth factors to grow and divide. They seem to make their own growth factors. They don't have a normal cell control system, nor do they exhibit or create what we consider normal cells. They tend to have small cytoplasm. They can have multiple nuclei. They can have very coarse chromatin. The cancer cells aren't making cells that can do anything other than just replicate. In terms of a cancer cell can be, so with that coarse chromatin that we just talked about, it can be very coarse, or so it can be genetically very different than the cell it started out being, or it can be just slightly altered. And so if a cell is just slightly altered, it more or less needs to stay in that area, and we call that a benign tumor. If in fact a cancer cell and its DNA is highly mutated from the original cell it came from, we consider that a transformation, or a transformational cancer cell. And that is the cell that actually can become a malignant tumor. Sorry, I can't speak. A malignant tumor. And a malignant tumor, because it's highly different than the original cells, can move from place to place, can metastasize. The immune system actually recognizes cells and can eliminate or get rid of cells if in fact it is being given enough time. I am not given enough time. I'm run out of time. So this is the end of chapter 12.